Good morning, all. Welcome to my presentation. Today, I would like to talk about ideal amino acid ratios or ideal protein ratios in relation to the dietary crude protein concentrations in broiler chickens. Why it is important? Because this relationship is certainly almost not what you think as it is. Our main objective of the AgriFutures project is to develop efficient reduced crude protein diets to the broilers. As you all know, conventional broiler diets contain more than 200 grams per kilogram of crude protein, as well as the crude protein digestion in feedstuff is not fully completed. Therefore, high amount of nitrogen can be excreted to the environment and can make severe environmental issues, as well as it can be negatively impact on litter quality and bird welfare. Moreover, reducing dietary crude protein also can minimize our reliance on imported soybean meal, and this can be a plus point of the context of country economy. Therefore, our aim is to reduce dietary crude protein in broilers without compromising their growth performance. However, broilers' responses to reduced crude protein diets is inconsistent. There can be many reasons, but as a team, we think that the feed grain we used in the diet is one thing. For instance, maize-based reduced crude protein diets perform better than wheat-based reduced crude protein diets. Moreover, there can be unbalanced starch and protein digestive dynamics in reduced crude protein diets. This explains the digestion of starch and protein, absorption of glucose and amino acid, and their transition to the systemic plasma through the enterocytes. Moreover, there can be deficiencies in non-essential amino acids in reduced crude protein diets, and they can have unbalanced dietary amino acid profiles. Therefore, we have to identify better ideal amino acid ratios for the reduced crude protein diets, or we have to modify existing ideal amino acid ratios to develop efficient reduced crude protein diets. Just a saying of Sir Isaac Newton, a great scientist: if I have seen further, it is by standing on the shoulders of giants. With this mind, I would like to talk about ideal protein ratio concept. This concept was firstly introduced at late 1950s and early 1960s at the University of Illinois and known as Illinois ideal protein concept. The underlying assumptions of this concept were the absolute amino acid requirements in broilers influenced by the dietary, environmental, and genetic factors, but the essential amino acid requirements relative to the lysine should remain largely unaffected by above-mentioned factors. Since the Illinois ideal protein ratio concept, there are many ideal amino acid ratio recommendations introduced by different scientists as well as different organizations. So in this slide, I tabulated some of the ideal amino acid recommendations since 1965 to 2014. If you closely look at these ratios, you can see that there are variation between amino acid requirements, especially on arginine, leucine, as well as valine. Moreover, you can see that many of the recommendations mainly focused on essential amino acids, but the exception was Wu 2014. He included all the amino acid into his ideal amino acid recommendation. Surprisingly, not many studies compare the impact of ideal amino acids ratios on broiler growth performance. So from the few studies, I picked Saliva et al. 2012. He compared 6 ideal amino acid ratios and obtained significant outcomes for weight gain and feed conversion ratio. So based on his results, feedstuff was the best ideal amino acid ratio. So based on this background, the objective of our project is to determine the impact of crude protein levels and ideal amino acid ratios on broiler growth performance. And we hypothesize that ideal amino acid ratios will have diverse impact on broiler growth performance depending on the dietary crude protein concentrations. So this study was conducted from day 14 to day 35, and we used ROS 308 male broilers, and they were raised in flow pens. In this experiment, we had 6 treatments with 7 replicates per each. 6 treatments consisted with positive control diet, which contained 230 grams per kilogram of crude protein and a commercial ideal amino acid ratio, and a negative control diet, which contained 210 grams per kilogram of crude protein and low density ideal amino acid profile. So we used whole body protein ratio as the low density amino acid profile, which reported by Wu 2014. Rest of the 4 dietary treatments followed the factorial arrangement, 2 by 2 factorial arrangement, which contained in 2 crude protein levels and the 2 ideal amino acid ratios. 2 crude protein levels were 210 and 180 grams per kilogram. And the 2 ideal amino acid ratios we used in this study, we would like to mention it as ratio A and ratio B. So these are the 2 ideal amino acid ratios we tested in this experiment. So as you can see, many of the amino acids are similar between these 2 ratios. CV values are less than 5%, but the exception was phenylalanine plus tyrosine. It have 9.08 CV value, and we think that this phenylalanine plus tyrosine concentration makes these ideal protein ratios more specific to each other. These are the dietary composition of each diet. I would like to highlight it here that once we reduce in dietary crude protein from 210 to 180 grams per kilogram, we have to increase the non-bound amino acid inclusion or supplemented amino acid inclusion nearly 3-fold factor. So this makes these reduced crude protein diets more different to standard crude protein diets. These are the nutrient composition of the diets, except the negative control diet. All the diet containing 11 grams per kilogram of digestible lysine. So we had less level of digestible lysine level in negative control diet because it is low density amino acid profile. Okay, let's move to the results. First, we analyze these 6 dietary treatment in 1-way ANOVA. And as we can see, there's a positive control diet clearly outperformed the rest of the 5 diets in weight gain and feed conversion ratio. And as expected, negative control diet is the worst diet. Moreover, you can see that broilers offered ratio A and B diets with 210 grams per kilogram of crude protein. It is not significantly different with positive control diet in weight gain, as well as in feed conversion ratio. Broilers offered ratio B with 210 grams per kilogram of food protein is not significantly different with positive control diet. In this study, we did not get any significant influence from the treatment for the feed intake. Next, we did the 2-by-2 factorial analysis for the rest of the 4 dietary treatments, excluding negative and positive control diets. The interesting outcome we got from this analysis, which says that under the 210 grams per kilogram of crude protein, ratio B is the best ideal protein ratio in both weight gain and feed conversion ratio. But once the crude protein level reduced to 180 grams per kilogram, ratio A beat ratio B. So this outcomes questioning the concept of ideal protein ratio, which says that dietary factors are not influencing ideal amino acid ratio and broiler growth performance. But our outcomes says that reducing dietary crude protein shifted the best ideal amino acid ratio from B to A. So why this happened? There can be many possibilities, but based on our experiment and the existing results, we think that differences in phenylalanine plus tyrosine into ideal protein ratios could be a reason. And also you can see that ratio B contain less phenylalanine plus tyrosine level compared to the ratio A. And based on the recent suggestions, which are Texas A&M and the Franco et al. 2017, they said that the recommended or the optimal phenylalanine plus tyrosine to lysine ratio is 105 and 112 respectively. So if you see, once we reduce in the dietary crude protein levels, in ratio B, phenylalanine and tyrosine level get below that recommendation. So we think that ratio B is deficient in phenylalanine plus tyrosine level in reduced crude protein diets. Moreover, there are some concern on the ratio between phenylalanine and tyrosine. The range is 1.22 and 1.22 to 1.33 based on Texas AM and the Franco et al. 2017. However, none of the other diets not following this ratio, but in reduced crude protein diets, ratio A and ratio B have a bit wider difference than than high crude protein diets. Supporting the fact that phenylalanine and importance of phenylalanine and tyrosine, we got some interesting relationship between some of the parameters of our experiment. Analyzed dietary concentration of phenylalanine plus tyrosine, linearly related to the weight gain, feed conversion ratio, breast muscle yield, and relative fat pad weights. Increasing this concentration improve the weight gain and breast muscle yields, as well as the feed conversion ratio. And increasing phenylalanine plus tyrosine concentration also reduce the relative fat pad weights. Instead of the ideal amino acid ratios, there can be some other confounding factors effect on the low performance of reduced crude protein diets. So in here, we got a quadratic relationship between starch protein disappearance rate ratio in distal jejunum and feed conversion ratio. So reducing starch protein disappearance rate ratio in distal ileum improve feed conversion ratio. So what this indicates? So as we know, glucose from starch digestion and amino acid from protein digestion, they both use sodium dependent transport system for intestinal uptake. Therefore, these two nutrients, glucose and amino acid, are competing for sodium for intestinal uptake. Therefore, we have to give correct blend of starch and protein in the diets, especially in reduced crude protein diets, as well as higher starch level can compromise amino acid digestibilities in broilers. The next confounding factor, we got a quadratic relationship between non-bound amino acid inclusion and feed conversion ratio. Increase in non-bound amino acid in the diet compromise feed conversion ratio. This indicates the importance of protein-bound and non-bound amino acid digestive dynamics. As we all know, non-bound amino acids are not undergo any digestion. They are available for the absorption. Therefore, increasing non-bound amino acid in the diet increase the percentage availability of amino acid in short time. Therefore, there can be some imbalances happen in the site of protein synthesis, amino acid imbalance. So these imbalances increase the deamination of amino acids, and this deamination increase the ammonia level in the blood. So the high blood ammonia level is not good for the animals. So they have to detoxify it and excrete it as uric acid. This whole process is the energy costing process, and it can negatively impact on the growth performance of the broilers. And we think that the quadratic relationship we obtain here may explain this rationale. Lastly, I would like to talk about whole body protein ratio. So we use this whole body protein ratio as the low density amino acid profile, and the birds perform poor. The bird performance was poor. So this indicates that the amino acid requirement for the functional purpose instead of the body protein synthesis. And also we think that whole body protein ratio is the base for the ideal amino acid concept, and we have to modify this whole body protein ratio based on the functionality of amino acids to obtain better ideal amino acid ratios. So in conclusion, yes, we got that conventional and reduced crude protein broiler diets demands ideal protein ratio. Based on our research outcome, it is phenylalanine and tyrosine. Also, when we considering ideal amino acid ratio for the reduced crude protein diets, we have to pay attention on starch and protein digestive dynamics as well as the digestive dynamics between protein-bound and non-bound amino acids. With this, I would like to conclude my presentation, and I would like to acknowledge AgriFuture's Australia Chicken Meat Program, as well as the University of Sydney and the Poultry Research Foundation, and at least but not last, the technical staff of our research group. Thank you very much. And I would like to get any question to answer from you.
Thank you very much, Shemil, for your presentation. And yes, we have some questions. Did you measure plasma amino acid concentrations in this trial, in particular phenylalanine and tyrosine concentrations?
Yes, we did analyze, and we got some, increase in phenylalanine and tyrosine level in the diet, increase the concentration of phenylalanine and tyrosine level in the plasma also.
Why did you start the experiment at day 14 post-hatch?
Normally, our research group mainly concerned on this grower phase, because we think that this is the most important phase, and we are dealing with especially non-bound amino acid inclusions. In commercial settings, non-bound amino acids are not much included into the starter phase from day 1 to day 13. It is not that much commercially viable, and we think that the grower phase is the most prominent phase, and that's why we are normally concerned on this phase.
Was blood ammonia concentration determined across the treatment groups?
No, we didn't. And also we think that it's not that much good parameter because of the previous research results. These ammonia levels can be variable, not only because of the treatment, because of the time of bleeding also can affect on these blood ammonia levels. And we think that it is not good parameter to determine the deamination of amino acids.
Were phenylalanine or tyrosine below the requirements during the experimental period?
Only in reduced crude protein diets, broilers offered ratio B is deficient compared to the recent recommendations.
Comparing the high protein diets, the reduced crude protein diets resulted in reduced plasma amino acid concentrations. Is that correct?
We cannot say like that because there are many confounding factors, and we also, every study we are analyzing our amino acid profile in the blood, and we didn't get that kind of outcome. It variables, it inconsistent based on starch level, the crude protein level, the level of non-bound amino acid inclusions. Therefore, we cannot exactly say increase in crude protein level, increase the plasma amino acid level in blood. Yeah.
Shemil, protein nutrition is very important for the starter phase. What diet did you use from day 1 to 13 post-hatch?
Yeah, we used a commercial starter diet, which contained 230 grams per kilogram of crude protein.
Thank you again very much for your presentation and for answering the questions.